Welcome you all on the behalf of Rahul Anand tutorial. So as you know that I am Rahul Anand and this is somewhat around my channel whose name is actually the Rahul Anand tutorial. So today I am going to deliver a lecture as well as some very interesting topic regarding the Aiims Neet as well as the All India Pre-Medical Examination, often the GC. So these questions are often being asked with reference to our examination. So let's talk about what does actually these means in the electrophoresis. So if I am telling you about the electrophoresis, so these are coming under the chapter of what? So these are coming under the chapter of the biotechnological principles. So as you know that. So what does DNA fragmentation actually mean? So what is actually the process of the separation of the DNA fragments? So let's talk about these. How is the DNA fragments are basically being distributed? So for that we are primarily having the electrophoresis technique or either the electrophoresis method. So what does it actually mean with the electrophoresis method of separation? So here electrophoresis method playing an important role for what? For demonstrating the value as well as for the separation of the DNA fragments. So often these DNA fragments are what? So these are created from what? So these are created from the endonuclease. So here with the restriction endonuclease. So what is actually the endonuclease? Often, so what does actually the endonuclease mean actually? So we are having the exonuclease as well as the endonuclease. So in this case of the endonuclease, we have the DNA fragments. So these are having the nucleotides. So if the nucleotides is basically being cut off from the middle or within the portion of the DNA, mean from the middle position, it will be coming under what? It will be coming under the endonuclease. But if I am telling you about the exonuclease, so what does it actually mean? So if the DNA fragment is being cut off from the side by side points, such as the beginning point or either the final point, so it will be coming under what? So it will be coming under the exonuclease. So you have got the concept of the endonuclease. So what does it actually mean? So it means that cutting the nucleotide from what portion? From actually the DNA fragment position within the DNA segment. So when we are cutting the nucleotide from within the portion of the DNA, it will be coming under what? It will be coming under the endonuclease. So these DNA fragments are coming from what? So these are coming from the restriction endonuclease. Now, so if I am telling you about the the electrophoresis, so what does it actually mean? So electrophoresis is itself composed of the two words. So what actually the two words? So electro plus phoresis. So electro means what? Something related with the electrons, something related with the charged particles, as well as the phoresis means what? Phoresis means it is having some kind of the cavity, some kind of the holes like structure which play an important role to abstract or particularly attach the DNA molecules, which are quite larger as well as the macro molecules in nature. So let's talk about the electrophoresis. Means something related with the electro. So the reference which is often having the pores like structure, playing an important role for what? To track down the larger molecules. Mean what? To track down the larger molecules of the deoxyribonucleic acid. So deoxyribonucleic acid. Now, so if I am telling you about the electrophoresis, the three things you have to remember with reference to these, mean the electrophoresis. So these are what? So these are actually the agar gel. Often we are having the two things such as the valves as well as what? As well as the electric field. So let's talk about the primary thing. Means what? Means the here agar gel. So what does it mean? Mean the agar gel. So these are the normal substances which are being abstracted from what region? It is being abstracted from the sewage region. So sewage means what? Something related with the hydratic solution. So hydratic solution is often having the different components. This is what? This is the alga. So alga itself is coming under these mean the agar gel. So agar gel is basically being abstracted from what region? It is being abstracted from the algal region and playing important role for what? Playing important role to filter down the macromolecules related with what? Related with the DNA. So DNA will be coming under the larger molecule. Why? With reference to these, mean what? Agar gel. So often we use the agar gel. Why is it so that we are using the agar gel? Instead that we are not using anything else. So this is due to because it is abstract. It is easy to abstract. It is easy to cast. It is easy to cultivate as well as it is easy to operate. So this is somewhat the usage with reference we are having the agar gel. Second is what? So agar gel. So these are coming under the agricultural case. So in terms of the agriculture, so we have talked about that it is being found from what region? It is being found from the seabed region, mean the algal region actually. So algal region. So alga is often been divided into three parts. So what actually these are the chlorophytes, pheophytes as well as the rhabdophytes. So what does it mean the chlorophytes? When something related with the chlorophyll. So chlorophyll means what? It will be having the green pigment like structure. It will be having something resemblance with reference to the chlorophyll pigment. Second is the rhabdophyte. It means something related with the red pigment as well as the pheophyte. So likewise, these are processes are basically being repeated on in the case of the algae. Thus, I often written for you the case. So agarose. So these are having the matrix-like structure of it. These are extracted from the alga, playing important role for what? For demonstrating, for cultivating, actually the huge amount to separate as well as to filter down the macromolecules regarding what? Regarding our deoxyribonucleic acid. Now, second is what? Second is the wells. So what does it actually means by the wells? So wells is having the pores-like or the cavity-like structure. So what does it mean that the pores or either the cavity-like structure? So these play an important role to give the storage accommodation for what reason actually? For giving the performance or either the station position regarding our mean the larger molecules with reference to the DNA, mean the deoxyribonucleic acid. So these wells are playing important role to attach those mean the DNA molecules which are quite macro in nature with reference to what? With reference to our mean the algal resin as well as the agarose gel resin. Now, so in the third we are having the electric field. So what does it mean that the electric field? With reference to this, in the case of the electrophoresis. So we are having the electric field playing important role for what? Playing important role to accommodate, to arrest down, to culture down, to accommodate as well as to mix up with reference to the external charges which are being produced in terms of the cathode, in terms of the anode. So cathode means what? Mean the cathode will be bearing the negative charge as well as the anode will be bearing what? It will be bearing the positive charge. So these are quite reciprocal cases. So you could often find here in the board actually. So here the anode. So anode bearing the positive charge as well as the cathode bearing the negative charge. So in this case, we are often having certain more things to go in. So DNA as well as the electric field. So it will be bearing the positive as well as the negative charge. So any kind of the attraction regarding that reason playing important role for what? Playing important for role in order for the repulsion process as well as for the attraction process. So it will be playing an important role to demonstrate the effort regarding that reason within the influence of the electric field. So in this often we are having the two facts regarding what? Regarding the electrophoresis method or rather the tagging. So our main achievement or rather the goal is what? To distinguish the larger molecules which are actually what? Which are actually the DNA fragments, mean the deoxyribose fragments. So as we know that deoxyribonucleic acid is bearing what charge? So it will be bearing the negative charge. So negative will be attracted towards what range? It will be attracted towards the opposite pole. So opposite pole will be what? It will be the positive charge. In the second we are having the PO4 3 minus actually. So PO4 3 minus will be bearing what? It will be bearing the positive charge. Thus it will be attracted with what reason? It will be attracted or either correspond with the negative charge or either the negative pole. So by this process means the DNA as well as the PO4 3 minus is basically being demonstrated. It's basically being separated off. So thus by this process we could get the accommodation. Thus by this process we could actually separate our DNA as well as the PO4 3 minus as well as the larger molecules as well as the macromolecules of it. So this is somewhat the case of the electric field. Now so after that we are having the process of the DNA fragments. So primarily what will be happening? Primarily it will start to break down, and in the second what will be happening? What? Can you tell me? So in the second it will start to separate down. Primarily breakdown, then separation, then what? Then the segregation process. So segregation means what? It will locate as well as go to the ideal position with reference to the ideal location, and hence by those processes we could get the ideal condition as well as the ideal DNA fragment is basically being segregated. So primarily it is basically being, we are having what? So it will be primarily breakdown, then what? Then the separation process, then the segregation process. Likewise you could often find here in the board. So the novel DNA fragment will be primarily isolated as well as it will be drawn. So isolated means what? It will be separated on. So separated, of course. Now, so let's talk about something more with reference to the electrophoresis technique or rather the method. So here, so the positive charge as well as the negative charge. So we have read about the DNA that it resembles the negative charge. As well as these are accommodated with reference to what? With reference to the opposite pole exchange. So after that we are often having certain things to go in it. What are actually these? So we are having the two types of the molecules actually. So what are actually the two types of the molecules? It will be the larger molecules as well as the smaller molecules. So as we know that larger molecules, of course. So we play it. So of course that if we are having the huge amount of the bricks, so it takes the amount of the effort to transport it within from one geographical location to the other geographical location. Regarding that, suppose that if we are having the cotton or either the small balls of cotton, so what will be happening? It will be easy to transport by us. So these are quite different. So this was the example to demonstrate you what. So demonstrate you actually the value with reference to these mean the larger molecules as well as the smaller molecules. So you could understand that the small molecules will deviate faster as compared to the larger molecules. It will take quite a bit of time. As like here. So when we are running, actually, so when we are walking, so we travel the short distance. But when we are using any kind of the vehicles, such as the aeroplane as well as the helicopter, we actually travel with reference to what? With reference to the quick geographical distance. So you could often find here. So here. So the smaller molecules will be traveling. What reason? It will be traveling quicker with reference to the smaller molecules, larger molecules actually. So larger molecules will play an important role as well as it will be transported, but with reference to what? With reference to the slow geographical rate as compared to the smaller ones. Now, so let's talk about the overview with reference to these with the electrophoresis technique. So primarily what is happening? So primarily we are having here, when the DNA fragments are basically being drawn. So from ideal locations. So DNA fragments are basically being drawn from here, mean the agarose. So it is being passed on from the agarose. Then what will be happening? Then we will be applying the electric field. So as we apply the electric field, these will start to radiate, these will start to cross bond, these will start to divert. So there will be the separation with reference to the larger molecules as well as the separation regarding the smaller molecules. So the smaller molecules will be traveling faster as compared to the larger molecules will be traveling slower. So this is what the method of the electrophoresis technique. So you would often find here. So I have often written it for you here. So if you like the work with reference to the education as well as the lecture, so you could often subscribe, like as well as share my videos with reference to what? With reference to the Rahul Anand tutorial. So thank you very much for watching the video. So if you like the work, please subscribe, like as well as share.